Hi guys, I am Dave and today we are going to talk about antibodies. In the past, 2 years we've talked so much about the SARS-CoV-2 and coronavirus and everything about the new vaccine that have been in development, and antibodies and immunity have been one of the very crucial part of it. Right, so today we are going to know basic about antibody and how actually antibody work. At this very moment of when you are watching the video, there are millions of antibody inside of you which are working recklessly, tirelessly to protect you against the pathogens. Okay, so now we'll talk about antibodies. So this you can see here that this Y shape is actually what called an antibody, and this is and here you can see that there are two pointy ends which are actually called the paratope, and these guys actually what they do is they interact, but interact with what? So for example, here we can see this is, let's say for example, a virus or a cell for example, which have protruding ends. So what happens that these protruding ends actually have affinity. What happens that these protruding ends actually have affinity, and depending on the antibody and the receptor, for example on this cell, what happens is that if they have high affinity, they bind with high energy, as compared to if they have low affinity, they bind with low energy. Right, and this is the cell's receptor. So this receptor is actually called the epitope, to which the paratope of the antibody binds. Okay, so, and this actually can do a lot of work by itself, and these are also important in like signal transduction. These are something which you'll more understand by going deeper into these scientific terms. Signal transduction in a general way means that how signals are being processed inside the cell. It can alter that, it can give a message to the cell, right? So now we are going to take a little deeper uh, view on the antibodies structure. So what actually antibody is? It is a combination of molecules in which there is a heavy chain and a light chain. So here you can see I have drawn two these are the heavy chains, and these are the heavy chains, and these are the light chains. And throughout the antibody, heavy chains though doesn't really change much, but light chain uh, changes a lot, and their name also suggests that why they are being called as a light or heavy chain is because of their uh, si- uh, because of their molecular size. Also, we should know that there are there are two types of chain. There are two type of domains actually. So here we see that there is a conserved domain. This domain usually get conserved because this is not the one really interacts with the antigen. This is an antibody. Whom does it interact with? It interacts with the antigen. So that's why we call this as an antigen, and it interacts to an antibody. So antigen is represented by Ag in immunology, and antibody is represented by AB. Right? So this type of interaction actually happens between the antigen and antibody, and this interaction is established by this. This is a very, very, very important part of an antibody, and it is called a variable chain, a variable domain of the antibody. Ever wondered why there are our body can defend upon to a millions and like a countless organisms which we cannot even see, right? So the reason being is this, the variable domain, because the variable domain has actually a lot of heterogeneity, right? So that is very sole reason of how we can defend against countless organisms, right? So here you can see this is the paratope of what? paratope of the cell, epitope of a cell to which the paratope is binding, to which the paratope is binding. This is an epitope of a cell to which an paratope is binding. This is an antigen, okay, and this can further cause the signal transduction or immunological reactions depending on what is the signal is being given to the cell. So this is the basic structure of the antibody. One more important thing, one more important thing to be noted here is that the heavy chains are being held by disulfide bonds. Also to note that these heavy chains and light chains are also held by disulfide bonds, right? So do study about this when you go through, if you're preparing for exam and watching this lecture, is that they hold by the disulfide bonds. Okay, so this is the basic structure of antibody, and there are many type of antibody that has been found in humans, right? Also one more thing, the two regions are actually called the Fab region, the Fab domain, fragment antigen binding domain region, whereas these domains are called as the FC domain, which is fragment crystallizable region. Reason being that these guys are the one who actually binds with an antigen. Meanwhile, this does not really have a function while binding to an antigen. Okay. Also remember that these are the N terminal, from where the antibody binds, via its paratope is called as the N terminal of it, whereas C terminal is on the downstream, is on actually the crystallizable fragment of the antibody. Okay, and now there are many types of antibody like IgG, IgA, IgM, IgD and IgE. You should know little about them. Is that first learn them in this order. It's called the GAMD, and these are in the increasing order of concentration. No, sorry. In the decreasing order of the concentration in humans. So IgG have been found in very high concentration, right? After that IgA is found, IgM is found, IgD and IgE has been very less, nearly 0.2% has been found in humans, right? IgG is one of the very important antibody, as it is highly secreted when there is a secondary infection, and even if it's a normal infection, after few days it has been seen that IgG is highly expressed, whereas IgA is the one who actually travels through the mother's milk, while when the mother is breastfeeding to kids, to their neonates in humans, right? And the IgM is the one who is pentameric, who have 10 epitopes to attach with. Okay, I will attach resources to this YouTube video which you can see. IgE is very special one. It is actually involved with the allergic responses in human. So this is some basic information about the multiple type of antibodies which have been found in humans. Now you can see that there are uses of antibodies beyond our own defense, right? So antibodies are also used in immunofluorescence, immunohistochemistry to elucidate. The immunofluorescence is basically used to check the translocation within the cell, right, of a protein or a molecule which is fluorescently tagged by an antibody. Immunohistochemistry also show the changes in the physical features, and ELISA, you know that ELISA is the enzyme link immunosorbent assay, where antibodies has been used to tag and check. ELISA has been used to check the presence of a particular antigen. Yes. So thank you for watching my video, guys. If you like, please hit the subscribe, and of course, uh, I'll make more videos about immunology. I will add resources below this video, so check it out. Bye. See you soon.